So today we have a really great speaker, Dr. Jean-Philippe Wert. He's right now a research scientist at Google Brain in Paris and adjunct researcher at PSL University, Mine Paris Tech. Previously, he received his PhD in mathematics from Paris University. He was research professor and founding director of the Center for Computation Biology at Mines Paris Tech, team leader at the Curie Institute on Computation Biology of Cancer, visiting scholar at UC Berkeley and professor of mathematics at École Supérieure in Paris. His research interest concerns the development of statistical and machine learning methods, particularly to model complex, high-dimensional and structured data, with application focused on computation biology, genomics, and precision medicine. Dr. Wert is definitely one of the most influential people applying machine learning to mathematics. Other than his seminal research work, there are also a number of excellent tutorial and videos lectures available online. Please check them out. They are really, really good. So without further ado, let's welcome Dr. Wert to deliver the seminar titled Computational Analysis of Tumor Heterogeneity from bulk to single-cell genomics.
Thank you, Jay. Can you hear me okay?
Yes.
Excellent. So thanks a lot for the invitation and the introduction. It's a pleasure to be able, to have these online seminars. We did not choose that, but it's quite convenient to talk everywhere in the world from your living room. That's what I do today. So I will, in the next hour, I will try to share with you some results and some projects I've been working on in the last few years related to computational approaches to study genomics, and in particular genomics of cancer. And I will focus basically on mostly one main project, which is inferring tumor heterogeneity from standard whole exome sequencing. I will explain a bit more about that. And then I'll spend some time discussing a bit the challenges and prospects of using single-cell genomics in this domain. If you have any question, feel free to ask during the talk. Because I'm watching my slides, I may not be watching the chat window, so don't hesitate to open your mic and stop me if there's anything you don't understand. Okay, so, we hear a lot about health these days. There is a big pandemic going on, but obviously there's not only the pandemics that affects our health, and cancer is still there and will be there for, at least the coming years. It's a devastating disease that kills 1 out of 6 human beings. It's not only in rich countries. And so there's a real need, still today to better understand and better treat cancer. So one question is, with all the progress in computational approaches, machine learning, etc., it's clear that we have more and more tools to maybe better understand the disease, the biology of the disease, and make prediction on how to better treat it. There are plenty of applications of machine learning for images, for example, for diagnosis, detection of cancer, identification of cancer cells from images. But what I will discuss today is specifically at the molecular level when we do genomics. So I suspect most of you are aware of what genomics is. I will explain a bit. But basically measuring information at the level of DNA or RNA proteins. And from there, trying to characterize cancer and hopefully in the future better treat it. So something which is well known when you study cancer, in particular cancer genomics, is that when you take different patients, often there are different diseases at the molecular level, right? So even though if we focus, for example, in breast cancer, in the common language, it's one disease. We say there is a cancer in the breast. But as soon as you look at which genes are mutated, which genes are overexpressed, etc., you may see that every single person has a different profile, right? And so the one big promise of precision medicine, personalized medicine, is to say that because every cancer is different, maybe you can treat them differently. So this makes complete sense, and there's lots of research on that front. So this is, the fact that different patients have different tumors is something that sometimes we call inter-patient heterogeneity, right? Different patients are different. Now, what I will discuss today is another level of heterogeneity, another level of complexity, which is that within each individual cancer, each unique patient, if you look closely at the cells that compose the tumor, then there is also lots of heterogeneity, right? And this is a bit an open question to characterize it, to quantify it, to understand it, and to understand its consequences. But to make a long story short, it's quite intuitive to realize that when a tumor grows, cell divides, and during the tumorigenesis process, so when the tumor grows, just like any development of cells, there is accumulation of random mutations. Then the mutations may be fixated in the population if they benefit the cells. And because it's a constrained environment with competition for resources, you can see appear subpopulations of cells, which we call subclones or dominant clones if they are dominant in the case of tumors. And so it's quite common that, on this plot, if you take from the left, which is the beginning of the cancer, to the right, which is the advanced cancer, when you take a sample, when you look at the tumor on the right, it's composed of different cells, which are not all the same, at least at the molecular level. And here I zoom, you can imagine the plots showing the chromosomes with some mutations. So if you look at the DNA of the cells, we may have different subpopulations of cells within the tumor, right? So this is just something that exists. And it turns out that, beyond the scientific interest of trying to understand this, there seems to be, and many people are convinced that there is a potentially large clinical interest to better understand this heterogeneity, in particular because it's likely to be frequently associated to resistance to treatment, right? And here, again, there is nothing conceptually hard to understand. It's just like in a pandemics, and just like what we witness now with the COVID-19, that you may have, I hope we will not witness exactly the same picture, but when you have a population, in our case, of cells in a tumor that starts having different genomes, and when you treat the whole tumor, which is what is shown in this picture, again, horizontally, it's a chronological time. In the center of this plot, you have chemotherapy, for example, that's given. So the treatment would kill most of the cells because it's designed to kill cancer cells. But it may be the case that in your population, there may be a minor subpopulation with specific mutations that is kind of protective against the treatment. And then once the other cancer cells die, then the minor subpopulation, which is resistant to the treatment, may survive and may have more room to grow. And so this may be the start of a second tumor, a relapse, which does not respond to treatment anymore, right? So this happens, this has been documented. It's unclear if it's always like that, but at least the appearance of resistant relapse of cancer is quite common. And it's believed that maybe if at diagnosis time we were able to characterize very finely the heterogeneity of the tumor, maybe identify potentially resistant subclones, we may be able to treat a bit differently the tumor in order to make sure that not only we kill the dominant clones, but as well we control the explosion or the future growth of the resistant subclones. Right, so this means that it's interesting and potentially useful to, if we were able, at diagnosis time, for example, someone with a cancer, to identify and to describe the population of clones, subclones present in the tumor. So how do we do that? I will, as I said in the beginning, I will mostly talk of one method that we've been working on with a colleague and student of mine called CloneSeq, which has been developed to try to infer the composition of a tumor in a superposition of the clones. And then I will discuss the potential use of single-cell genomics, which seems to be well adapted to answer these specific questions. All right, so let's start with bulk sequencing. So by bulk sequencing, I mean the opposite of single-cell sequencing, meaning that this is the standard sequencing or standard genomics, where you take, typically, if you look at the picture on the right, you have someone with a cancer, and if you want to make the genomic profile of the tumor, you would take a sample of the tumor, like a biopsy, put it in the machinery of sequencing, etc., in order to get the DNA, so to read the DNA in the tumor. Then you may do that. You may do the same process on some normal cells of the person, like taken from blood. Read the DNA, so you get the DNA of the person on the left, the normal DNA, and on the right, the DNA of the tumor. And by comparing the two, you may detect differences, which would be the mutations specific to the tumor, which are called somatic mutations. So I will just focus for the moment on reading DNA, even though I will talk later a bit about other measures you can do, like gene expression or epigenomic marks. But for the moment, I will mostly talk of DNA mutations. So on this picture, it seems simple, like you can measure somatic mutations, meaning it allows to separate mutations which are present in the tumor versus mutations which are absent from the tumor, so present in the normal DNA of the person. But as I said before, if you expect, so if you have a tumor which is made of different subclones, then the definition of mutation is slightly more complex, because if I come back to this picture, you see on the right that you have mutations which may be common to all the cancer cells, basically the mutations that occurred early in the tumor, so the mutation in the orange cells that were given to all the descendants of them. But you may have mutations specific to subclones, like the purple cells or the blue cells, which would not be present in all the cancer cells, but which would still be present somewhere in the tumor. The question is, when you sequence a tumor, what do you get, right? You don't really get a binary signal for each possible mutation, but maybe something slightly more complex. And maybe there is some information there that would allow you, just by reading the sequencing, to reconstruct the composition of the tumor into subclones and maybe associate each mutation to one of the subclones. So actually, this is exactly what happens. Here I need to go a bit, not much, but a bit deeper in how sequencing is done. I assume most of you know that, but just in case, when we say we sequence, a sample like a biopsy, it means that basically we randomly pick some DNA from the sample and read it. So we randomly pick a small piece of DNA, we read it, this is a read, and then based on the read, we reconstruct the genome. And so you have to think that if you have a sample which is made of heterogeneous DNA, like if you look at a specific position that could be mutated or not, then when you read a random piece of DNA in your sample, depending on where the random piece comes from, you will read either a mutated read or a non-mutated read. And the proportion of mutated read will be, in theory, related to the fraction of DNA in your sample that is mutated. So if we forget for a moment about the fact that cancer cells may have a varying number of chromosomes, if we just imagine that all cells have, for example, 2 chromosomes, basically the fraction of mutated reads would be an indication of the fraction of cells in your sample that contain the mutation. And so based on that, so here, it's just a cartoon illustration of what you can expect. If you take a mutation that is present in the orange cell, so an early mutation of the tumor, for example, then that mutation will be present in all the cancer cells. So you would expect the frequency of the reads of that mutation to correspond to the frequency of cancer cells in your sample. Now, if you take a mutation which is associated to a very late event, like which is only present in the blue cells, then the fraction of mutated read would somehow correspond to the fraction of blue cells in the population. Okay, so basically each mutation, if you just analyze the number of reads which harbor the mutation versus the reads which are not mutated, gives you an indication of the fraction of cells that have the mutation. And so because we expect the different colored cells to correspond to different populations, it's a bit like speciation, like the DNA is a bit fixed. Then you may expect many mutations to have similar frequency, like you may expect many mutations specific to the orange state, many mutations specific to the purple state, and many mutations specific to the blue state. And therefore, one way to identify the presence of subclones and their frequencies is just to take all the mutations you observe in your tumor, for each of them to compute the frequency. So there is some processing here because, as I said, you need to take into account the fact that you may have more than 2 copies of DNA, but I will not talk about that today. So assume you are able to process the count so that you have an estimate of the cancer cell fraction. Then if you plot all the cancer cell fractions of all the mutations, you may obtain something like what is shown on the bottom right here, which is histograms of frequencies. And here you see you have different peaks in this distribution. Some peaks on the left corresponds to cancer cell fractions around 1. So this would correspond to all the mutations in the orange, that appeared at the orange at the time of the orange clone, which with a cancer cell fraction of about 1. By convention, 1 means that it's all the cancer cells which have the fraction. And then you have a second peak and maybe a mixture of second and third peak corresponding to the some lower fraction mutations specific to the purple cells or the blue cells. Okay. So in this cartoon, I gave you the composition of the tumor and the corresponding picture that you get. And now, if you don't know the composition, but you want to infer it from the reads, then you need to do the reverse process, which is you start from all your reads, you compute the cancer cell fraction, you make the plots, so these histograms. And then the question is, from these histograms, can you guess that there is not a single peak, but here that there may be 2 or 3 peaks? Can you estimate their frequencies? And can you associate each mutation to each of the peaks? This would correspond to estimating the presence of subclones, the frequency of each subclone, and the mutations specific to each subclone. Right? So stated this way, I simplify a bit the question, but stated this way, it looks like a pretty simple question. Like you have this distribution, and the question is, can you fit it with 1 peak or 2 peaks, 3 peaks? Maybe you can fit a mixture of Gaussians, and the mixture of Gaussian-like thing would give you, the number of peaks you have, which is the number of clones and their frequencies, etc. It turns out that it's not, as soon as you go to the details, there are many variants of the ways to formalize the problem, and many people have thought about that and proposed method. And it's funny. So I take this plot from the PhD thesis of one of my students, Judith Ducassis, who just made a big literature review last year of methods that were developed to solve that problem. So basically, to go from this histogram to an estimate of how many clones, frequencies, etc. And so in this table, imagine that each column, so you have many names on the bottom, which are hard to read. These are different algorithms that were proposed in the last few years. So I think there are about 90 of them here, and it's still growing. I will not detail at all this picture. The goal is just to show you that there are many people who have thought about that problem, who have proposed different solutions. The method, they vary in how they formulate the problem, in what they take exactly as input, if they take the mutations, copy number, etc., if they work on whole exome or whole genome, etc. But there are plenty of methods. So this suggests that the problem maybe is not completely solved if there are so many methods, and I'm not sure there is one of them that's considered the one that works. Like many people use different methods even today, and so so one question is based on that before inventing. So one message is if you're a student in the room, maybe it's not a very good idea to think that working on that topic is good topic because it's so crowded yet. But one question is, which method work, which method don't work, what's the state of the art in this domain? Actually, it's hard to answer because most of the publications, there is not I'm not aware of any big benchmark comparing all the methods. We did one benchmark. I will go quick on that. This was published 2 years ago, comparing just a few methods in terms of taking TCGA, so samples, and even though we don't know the ground truth, at least we can compare the predictions of different methods. And what we found, to make a long story short, is that overall there is very little reproducibility between methods. So if you take 2 different methods, you get 2 different results in many cases. And even for a given method, depending on how you select the mutations that you show to the method, there are different things called the protected, public mutations. It's just which subset of mutations you give to the method. The method will give you different answers as to whether there are subclones or not in the tumor, what's the frequencies, etc. So I will not go, it's not so interesting maybe, but the take-home message is that I think it's still a widely open question as to what the good answer is. And certainly the question itself is, to some extent, ill-posed. Maybe if you go to too many details, the question of there may be an infinite number of clones if you did, each cell could be a clone if you go to too many details. So maybe different methods have also different notions of what is a subclone. Okay, so still we wanted to, we thought about, would it be possible to bring something to the table, a new idea maybe to try to improve the accuracy. Something you notice, maybe I come back here, is that you see that here, one difficult setting is when you have two subclones with similar frequency. So for example, here on the bottom right histogram, it's quite easy to see that you have at least two peaks, which correspond to two, so a dominant clone and a subclone with very different CCF. CCF is the cancer cell fraction, is the frequency of cells. So when you have two well-separated peaks, it's easy to separate them. But sometimes you have two peaks, like here, the purple and the blue, who don't have a very different CCF, and therefore you see that the signal-to-noise ratio is quite bad, in a sense that it's hard to say whether there is just one peak or two peaks here. Is it one clone or two clones? Just based on CCF, you see that if the CCF is similar, it's hard to differentiate different subclones. So one question we asked is, would there be some other information in the data beyond the CCF that could help us guess the presence of subclones? And so we propose to look at something else, which turns out to be another big thing in cancer genomics, which is the notion of mutational processes and mutational signatures, which somehow says that in addition to looking at the number of reads which are mutated, it may be interesting to look at the sequence of the reads. So when we talk of a mutation is something like a C was mutated to an A, and you may look what it is surrounded by. Okay, so if you look at that, each mutation can be classified, for example, by the which nucleotide is mutated into which, and what is the left and right nucleotides. And because we have two copies of DNA, so DNA is a double strand, we cannot differentiate between a mutation, so a triplet and a mutation, and its reverse complement. So in total, if you count how many types of mutations, and by type I mean something like A followed by a C that was mutated to A followed by an A, then there are 96 types of possible mutations. Right? So this means that literally when you take the sequencing machines and the data that you get from the sequencer, and you just focus on the mutations, then for each mutation you have one of the 96 types, and you have the number of reads which are mutated and the number of reads which are not mutated in your biopsy. And so looking at the types is something that was proposed by several people. In particular, there is a very nice series of papers by Alexandrov and colleagues who showed basically that, long story short, if you take, for example, different cancers, you take lung cancer, bladder cancer, etc., and you look at all the mutations and at their types, then you see clear differences in the types of mutations in different cancers. In fact, there's been lots of work on trying to model these differences in signature, in mutation types. And so Alexandrov and colleagues proposed the concept of signatures, which are basically what you see on the right. These are distributions, categorical distribution over 96 types, which were inferred automatically from analyzing large collections of tumors in order to summarize the patterns of mutation types observed in different tumors. And somehow using the concept of signatures, they were able to characterize each individual tumor by a mixture of different signatures. And the thing which is interesting is that not all, but many of the signatures have been linked to specific environmental or biochemical processes that induce mutations. Like exposure to UV ray creates mutations in DNA, but preferably of some types. Tobacco consumption also generates mutations, but of different types. So if you, and also, for example, if a DNA repair mechanism is broken, then it will create mutations also of some different types. So the different signatures correspond most of the time to specific processes, and you recognize them in the tumor. So typically when you have a tumor, just looking at the mutations and the types gives you information of why, what process was driving the tumor growth. It could be because of tobacco, or it could be because of mutation in BRCA1, etc. All right, so what's the link with ITH, intratumor heterogeneity? What we propose to consider is the following hypothesis, which is just an hypothesis, it's not always true, but it's the fact that maybe in some cases, when you have tumorigenesis, so when the tumor grows and you have some clones that appear in the sense that a subpopulation becomes fixed in the population because it has a set of mutations that have an advantage, etc. We propose that maybe in some cases, the change, so the creation of a subclone is accompanied by a change in mutational processes, and more specifically, a change in the signatures which create the mutations. Right. So typically, to be more concrete, imagine that a new clone appears because at some time DNA repair, so a gene, a protein involved in DNA repair is mutated and works less well. Then you will expect to suddenly to have more mutations appearing in a subclone, specifically on a subclone, and this new mutation would be due to the defect in this DNA repair mechanism that would be linked to some signature. Right, so this means that the new signatures in the subclone maybe would have different patterns in terms of mutation types than the mutations before this event. Right, so this would be a case where you would expect different mutations in different subclones. Maybe a similar example is in lung cancer. You could say that the first clone due to tobacco consumption would have the signature of tobacco. But then you would have subsequent mutations that would induce DNA repair mechanism defect, etc., that then would not be driven by tobacco, but more by other biochemical processes. Okay, so the question is that an hypothesis that that we can validate or not. So I don't have any biological result, but I can show you examples, real data that suggests that indeed in some cases we have different subclones. So we have a male clone and then a subclone. And we also see that the mutations are a bit different between the two clones. So this is an example here of one sample taken from the Cancer Genome Atlas. This is public data. It's a sarcoma. There are 246 mutations. So this was obtained by what's called whole exome sequencing. And so here on this plot, I just plotted the 246 mutations as plots, so as points in 2 dimensions, right? So each plot here with the color is a mutation. The 2 dimensions correspond vertically to the cancer cell fraction. So it's the thing that I talked about before. It was the fraction of cells that contain a mutation. And the horizontal axis is the mutation type. Again, for each mutation, we just look at the sequence and we say, is it, for example, A followed by A mutated to C followed by T? This would be one of the 96 mutations. So what do we see here? We see on this sample that quite clearly, if you just look at the vertical axis, which is, I plotted the histograms, the corresponding histograms on the right, we see clearly that there seems to be 2 peaks, at least. One peak around a CCF of 1, so the green thing seems to be the dominant clone, the mutations which are present in all the cancer cells. And then there is an accumulation of mutations with a CCF around 0.4, which suggests that there is at least 1 clone at 40% frequency in the tumor. Right? So in this case, it's easy to see that there are 2 clones. But what I want to highlight in this picture is that if you look on the horizontal axis, so what are the 96 mutation types, just by eyes, you see that there seems to be a different usage of mutations, and in particular on the top picture, so on the accumulation of cells with a cancer cell fraction of 0.4, you get many points in the C mutated to A set of types. Whereas if you take the mutations in the dominant clone, so with the CCF around 1, you have, visually at least, you have an accumulation of mutations in a C mutated to T bucket, right? So this is a case where there seems to be 2 clones, which we see very well from the CCF only. But interestingly, the 2 clones have different types of mutation. So this suggests that our hypothesis that somewhere we have a new clone and a change in mutational process occurs, at least in this sample. There are many more examples we can visualize, but this suggests that this exists. Now, why is that interesting? Well, this could be interesting for one thing, which is to identify subclones. Because remember that I said that most of the, I showed you the big table with around 90 methods, the majority of which just consider the information that you see on the density plot, right? So this was the histogram of CCF. And here what you say is that instead of just looking at one axis, if you look at two axes, so mixing the cancer cell fraction and the mutation type, there may be two sources of evidence that you have different subclones. And in particular, imagine that the CCFs of the two clones were close to each other, then it would be hard to see them from the histogram only. But by looking jointly at the histogram axis and at the mutation type axis, we may be able to better see the presence of two subclones, just because basically, it's easier to discriminate, to see two accumulations of clones in 2D than in 1D, basically. So this raises the question of, can we do that? Can we, try to make a method that detects these clusters of mutations, not only on the vertical axis, but also on the horizontal axis, and horizontal just means in the space of mutation. All right, so based on these ideas, we just did that. So this is a joint work, mostly the work of Judith Abecassis, who did a PhD I mentioned earlier, and a colleague of mine, Fabien Royal, who is a medical doctor at the Curie Institute. And so I will not go to any technical detail. I'm happy to take questions, and we have a tech report online, as well as the method you can use it. But basically, if you're familiar with what's called graphical models, this is just a graphical model where what we do is that we say we observe some data. So remember, for each mutation, we observe the mutation type, as well as the number of mutated reads and non-mutated reads for that mutation. And we model these fitting at 3 random variables, which are observed. So on this picture here, each circle corresponds to a variable. The darkened circles, so BN, DN, TN, correspond exactly to number of mutated reads, total number of reads, and TN is the type. So we say these are random variables. And the thing we add to that is that we add some correlation between them through some hidden variables, which would be here UN and SM, which correspond to the clones. Right? So we say that there are some clones that we don't see, but conditionally to the fact that we know that the mutation comes from a clone, then the clone generates some distribution over CCF and some distribution over trinucleotide context. So it's a kind of simple and natural model. And then we just use standard probabilistic inference tools to say, if we don't observe the hidden variables, which are the clones and the signatures, etc., then we can infer them using maximum likelihood and then conditional inference of these graphical models from the observed data. So I will not say more, but again, the software implements all that. And so what you get at the end is that for one sample, you give to the clone C the set of mutations, their frequencies, their types, and the software infers how many subclones there are. For each subclone, it infers what are the signatures which are active. So here it uses the language of signatures proposed by Alexandrov. There are like additional signatures. So we say that each subclone is a mixture of these signatures. This is some latent variable. So this is inferred automatically. And then something else that it does is that for each mutation, we have thousands of mutations. Each of them, we have the conditional probability to belong to each of the clone and to be generated by each of the signatures. Okay, so we try to assess if it works. It's a bit hard because this is a domain where we don't have much ground truth. We want to process real data, but on real tumors we can observe tumors, but we are very as far as I know, all the publications I see are do not know exactly what the answer is, what's the ground truth. So what we did is first to do simulations, and then to test the method and try to quantify if it works or not, and what are the parameters that influence the performance. And then I would show some application on real data, but not to validate that it works, more to try to start doing some research on the real data. Okay, so on simulations, I will not spend too much time because it's lots of results, but basically, just for you to know, there are many models that have been proposed to simulate mutations. Just like I said, there are many methods to infer ITH. There are also many methods to simulate data according to different models. So we picked what we found, like 4 different models. We compare to, in this case, 8 other methods for ITH inference. And when you do inference, you infer many things, right? You infer the number of clones, you infer for each mutation to which clone it belongs, you infer the CCF of each clone, etc. So it's hard to summarize all that in a single number. So here we followed some community-defined measures, which, in fact, there was a DREAM competition on that topic. So we followed the DREAM competition way to assess the accuracy, to see if a method works. And there are 4 scores here to assess the performance. So long story short, here you see radar plots. So each plot shows you on the 4 accuracy scores how different methods perform. So if you're outside, you're better. If you're near the center, you're not very good. There are not huge differences between the methods, right? But if you look on the right, so there are 3 plots on the right that correspond to simulation where our hypothesis is met, meaning that we simulated data where there are subclones, and we expressed, we made the simulation so that in different subclones, there are differences in signatures, mutational signatures, right? So this is what I call a favorable scenario, in the sense that this is a scenario in which our method is supposed to have an advantage because it takes into account this difference in signatures to increase the power in detecting subclones. And so if I don't have a very good visualization here, but if you look, for example, at the first radar, you see that the orange dots tend to be outside. So the orange is CloneSig, is our method. And here they significantly outperform all the other ones. And in fact, in all the 3 plots, they're winning on all the scores. So, this is our method. I show the results of my method. There may be some bias, but at least on the results we obtained, CloneSig seems to be working well in the case where, indeed, we simulate data with differences in CCF and differences in signature. On the left, the 4 plots correspond to cases which are not favorable, in the sense that here we just simulated data using existing simulators where there is no difference in signature. So these are cases where we don't expect CloneSig to have any advantage compared to other ones. And what we see is that there is no big difference, but CloneSig basically works at the level of the best method. So the take-home message from these simulations, and these are just simulations, is that the method based on the graphical model behaves basically like state-of-the-art methods when there is no difference in mutation types between subclones. And as soon as there starts being differences, the performance of CloneSig improves, and this continues. All right, this I will probably skip because this is more detailed results. Maybe the only comment I will do here is that we checked, because these are simulations, we checked the performance on the different axes of the different methods when we change different parameters. The only thing I would say is that one important parameter is the number of mutations you use, because in practice, especially in hospitals, when we sequence tumors, I think that most of the sequencing these days, and just because it's cheaper and non-convenient, is what's called whole exome sequencing. So we just sequence the exomes, so the coding DNA, which is just a few % of the whole DNA, as opposed to whole exome, whole genome sequencing. Right? And so if you look at the mutations that you get, of course, you get much less mutations when you just focus in the coding regions of the DNA than in the whole DNA. And on average, I would say that it's fair to say that when you focus on cancer, it depends on the cancer types, but in whole exome sequencing, we expect to have a few hundred mutations, like the sarcoma I showed before was 246. Whereas if you do whole genome sequencing, it's common to have 5,000 mutations, typically, right? So if you want to be able to use a method on whole exome sequencing, you expect it to work with a few hundred mutations. And if the method only works with a few thousand mutations, which is better because you have more data, then you could apply it to whole genome sequencing, but not to whole exome. So this is a big difference. And so here we just mentioned that the central column here shows the performance as the function of number of mutations. So this is how many mutations you see. The orange curve is clone-seq, so it tends to increase a bit with the number of mutations, but it still works reasonably well with 100 or 300 mutations, which is the standard that we expect in whole exome sequencing. I would just mention that there is another method which is called TRACK-seq. So I should have mentioned it before, maybe, because I said that we propose to consider mutation types and CCF, and in fact there is at least another famous method called TRACK-seq that does the same. So it also says, so TRACK-seq and more recently TRACK-seq-FREC are methods that also take into account, so try to do ITH and to model some changes in mutation signatures. However, so the methods are different. I will not go to details, but the method, so TRACK-seq has been developed to handle whole genome sequencing data. So cases where you have a few thousand, typically 5,000 mutations, and not for whole exome. It's written in the paper, but still we tried it here. And so what we see, so TRACK-seq is the TRACK-seq and TRACK-seq-FREC are the bluish curves. You see that indeed when you have 5,000 points or 5,000 mutations, it tends to work quite well, depending on the measure you look at. But as soon as you go to 600 or less than 1,000 mutations, so the range of whole exome sequencing, the performance degrades a lot because fundamentally it's a method that requires enough mutations. Right, so a big difference, I would say, between clone-seq and TRACK-seq, which are the two methods that try to infer changes in signatures, is that clone-seq, just because it's designed differently and it's just a graphical model that does not require a specific number of points. So clone-seq seems to be working on simulation for whole exome sequencing data. Okay, a couple more comments. So, I told you that we tried either to make differences in signature changes or not. And here this plot summarizes a bit the fact that, the more difference you have in terms of signatures, and the easier it is to detect subclones which have a small CCF difference. In this plot, vertically, you have the CCF difference. So if two clones have similar frequencies, I told you it's hard to separate them. So for example, here on the left plot, on the left column here, you see blue is bad, brown is good. So here you see that around 0.29 or 0.42, you move from good accuracy, bad accuracy to good accuracy. So you need at least a difference of, let's say, 0.4 to detect two subclones if there is no differences in signatures. And when you go to the right of this plot, then you increase how the signature is different, so how the mutational processes are different between the clones. And you see that suddenly you manage to differentiate between the two clones, even though the CCF difference may be smaller. So basically moving from 0.4 to 0.2. So this is just a quantification empirically of how using 2 dimensions is better than using only 1 dimension when there are differences in the 2 axes. Okay, last thing. So we tried the method on real data. So here, I cannot say if it's good or not because we don't know the ground truth, but at least, as I said, we can apply it to whole exome sequencing. So we did that on a big cohort in the Cancer Genome Atlas, which allows us, for each type of cancer, this is just a snapshot, and some like BRCA is breast carcinoma, we detected the presence of subclones in about 1/3 of the samples. This may be true or not, but this is our best guess. So this means that subclone, according to our model, is quite present in the real samples. And so these plots, I will not zoom on it because it's too technical, but they try to not only to say there are subclones, but then to see, between the dominant clone and the later subclones, what are the signatures that either disappeared or appeared, right? So that to try to make a story about, okay, we kind of try to infer the history of the tumor, saying that initially the tumor started with these signatures active, and later there was a subclone that occurred, and suddenly new signatures became active and old one became inactive, etc. Maybe I'll just, for example, illustrate that on one of them. If you take skin melanoma, this is the simplest example, I guess. But we see the presence in this, so this is skin cancer. We know that one signature called signature 7, according to Alexandrov, is a clear signature associated to UV light exposure. And so what this plot shows is that over most cases where we see that there is a dominant clone and then a late subclone, then signature 7 here on the left, so the bar on the left means that the signature 7 have decreased, meaning they were present in the dominant clone, and then in the later subclone, they disappeared. So, it makes sense that this was just totally obtained from the data, but this corresponds to a natural story, which is that melanoma starts because of UV light exposure, and then when it starts expanding, you have other processes that create mutation besides the UV light exposure. We have similar stories for the signature 4, which is tobacco signature in lung cancer, that is also present initially and then becomes much less active in the subsequent subclones. All right, so I mean, if some people so I'm not a medical doctor, oncologist, so probably I will have difficulties to go to more details if you have questions, but we can discuss that later if you're interested. And of course, we have a paper with a much deeper analysis of the different findings that go a bit beyond these trivial examples, but useful examples. Okay, so I will now turn to the second part, but as I said, this will be shorter. I just want to mention that, of course, if you're interested in heterogeneity, then it makes sense to look at single-cell genomics, because it seems that single-cell genomics is the tool in order to measure, visualize, assess heterogeneity. Just reminding you, probably you've heard, Jay is here, and probably many other people in the team know this data, but basically, long story short, the main difference between bulk and single cell is that if you have a tumor, so if we use that for tumor analysis, you start from a tumor, you put it in a machine, the protocol is different, but what you get instead of one profile, like the average mutations, is a per cell measure. So in this plot, maybe this is more for gene expression. But instead of a column, you get a matrix, right? So the sample is not just a vector of gene expression in this case, but it's a full matrix where each column is a cell and each row is a gene. And therefore, if you, for example, visualize the population just for one sample, you may see appear some populations of cells which are similar. So this could be done at the level of mutations or the level of gene expression or epigenetics, etc. But clearly, it seems that here there is no need for deconvolution because you already have the measure at the most basic unit level, which is a cell, and then it's more a question of aggregation here than starting from the average and deconvolving into subgroups. Okay, so it's starting to work on single cell. I would just mention a couple of work we have done, and I will not go to any detail because I don't have time, but I'm happy to chat later. So I just mentioned that to date, I mean, single cell, if you've never worked with them, you should look at them. This is super exciting. There are more and more data. There are some challenges, like typically because we measure, the genomic content in each individual cell is so small that, for example, when we look at data, we have plenty of zeros. This is a bit strange. First time you see that, it's mostly, matrix of single cell data is mostly zeros. So this means that you need to do something to analyze them. There are statistical properties like overdispersion, etc. So, a couple of years ago, when I was at Berkeley with Sandrine Dudois and colleagues, we worked on a model which was asking a question: suppose we want to do PCA on this data, but doing PCA on such a matrix, and you can do it, but the data are far from Gaussian, the noise is not Gaussian, etc. So how do you adapt this method to this data? So we did some model where, instead of PCA, we did the equivalent of PCA, meaning there is some underlying low-rank projection of your data, so meaning that each cell is projected to a low-dimensional space. But then instead of saying that the matrix you observe is just the product of a scoring and loading matrix like in PCA, just say that the PCA matrix defines the parameters of a statistical model, which in this case is something called the zero inflated negative binomial, just to account from the many zeros and the overall dispersion. And then by fitting into the data, it's a way, to fit a model to the data, which are a bit strange, but still recover a low-dimensional view of your cells these days. Right, so the method is ZimiWave. Again, it's I think it's a bit old now, even though it's still working. And using that is a way to. I mean, we can show that it's a way to be a bit more robust to batch effects, to have better. Once you have used such a model, to have better lineage reconstruction, for example, or to be more robust to the dropout. So that's all. ZimiWave is something a bit old, and something interesting that this is a very hot topic. I think after, in the last 2 years, there has been an explosion of methods that take different strategies, but to go from the raw data, so single cell, in this case RNA-seq, and learn some representation of cells in low dimension, using, for example, deep learning techniques like autoencoders, variational autoencoders, etc. Some work we did recently with, again, colleagues at Institut Curie in Paris, and this is part of the work of Felix Raimundo, PhD candidate, is to say, well, there are so many methods, so again, which one should you use and how easy are they to use? And for those of you who are familiar with deep learning, so Google, so everybody is doing deep learning on images, text, etc. But in fields like in images, I think it took quite a few years for the community and it's a big community to converge to some architectures and techniques to train the models, learning rate, dropout, etc. So the community in image is big, and it took some time to converge to a set of methods that now everybody uses. And now it's dangerous to change anything, right? If you want to make a deep learning for images, you should use a ResNet or one of the pre-trained model, and then it works. Otherwise, you may get very different results. And so our feeling was that, the same trend appears in genomics. So many people are developing, for example, deep learning models for genomics, but the community probably has not yet converged to the right architecture. There hasn't been, competitions or ways to make sure we have the good things. And so what we did is we did the benchmark using pseudo-simulated data. So, starting from real data, but making in silico mixtures of real data, and asking, well, if you have the data and you learn use one of these embedding techniques to embed the cells, can you recognize the cell tags, for example? So I still say this was a big work because we compared 5 methods, but varying all possible parameters. Like if you download any of these, any of this model, you can use it with default parameter, and you also, but you can change the parameters. In general, when you use the default parameters, it doesn't work very well. You would be surprised. So in this plot, again, I will not. It's a bit overcrowded in this plot, but you have a set of bars here. So each set of bars is one benchmark. So we created many simulated real data benchmarks. If you're high, it's good, and each color corresponds to a method. And I'm sorry, I forgot the legend of who is who. But the important is that, for example, if you take a challenging data set, so here, I'm sorry. If you take the fourth of the group of columns, you see it's a bit lower than the others. So you see that all methods are not so good because the vertical, if you're high, you're good. And you see that you have a full columns and dotted columns. So you have some performance which doesn't go very high, and then each bar goes a bit higher with different lighter colors. So the full color is when you use default parameters. So this shows you that for different methods, if you use default parameters, you're quite far from the best you can get from the method. And then when you do optimization and a posteriori choose the best parameter, you get better performance. Okay, and so the main message of this study, which is summarized in the paper, is that basically it's not completely clear today what method I would advise to use, and in particular, it's clear that let's say the easy methods, which would be PCA. So some methods like Scran and Sora, which are super popular, basically use PCA to do that. Then you have ZBWave, which I mentioned, which is kind of easy as well. So the model is more complicated, but there is no free parameter. You just need to say what's the dimension, and then it does all the things. So these methods are quite robust and give good results, on all our benchmarks. And the deep learning-based models. So here we just tried DCA and SCVI. These are two examples of some autoencoder and variational autoencoders. We observe that they can give very good results, but not with the default parameters at all. And the thing is, it's not easy to know how to tune the parameters, because if you don't know the ground truth, you can just play with the parameters, but you don't know, there is no clear measure to optimize, to tune the parameters. So I think, I'm sure it will change in the future where more people use them, when we have more data sets and probably bigger data sets. But for the moment, I think there is a clear potential for these, nice deep learning models, but they may be hard to use in practice just because, if you just, what we try, if you just use them with default parameter, you get often crappy results, and it's not clear how to find the good parameters in this case. All right, I will maybe almost conclude here, just say that there are many other fantastic challenges in single-cell data. One of them is how do you integrate different types of data? I talked of mutations, later I talked of gene expression. Now we work on epigenetics with the Curie Institute to try to follow breast cancer progression in terms of epigenetic marks at the single-cell level. One important question is how do you combine all this data? And I will not spend time on that because we did some work with Bill Noble and colleagues, but this work, which is super nice, was mostly done by Jay. So if you want to know more about that, I was happy to collaborate with Jay on that. And I suggest you ask Jay if you want to know how we propose to integrate heterogeneous data. All right. This I will just keep it. It's a bit different, but I just wanted to say that another direction, which I think is interesting and for which there are many open questions we don't know how to do, is how do you infer dynamical processes? So when you have single cell, you visualize individual cells, and sometimes there is a process, time process that could be differentiation, that could be tumor progression, that connects the cell. So a nice question is, can you just from single cell data reconstruct the dynamics, maybe infer gene regulation, because if you can, create a dynamical model, then gene regulation should be one of the factors that says which gene influences the future evolution of which other genes. I would just say that we worked on that. We have, we propose one method that tries to fit the dynamical model on data. I'm not sure I can spend much time on it, but it's a standard optimization problem where we say we have many cells. For each of them, we have a vector of gene expression on the one hand. And if we were able, in addition, to have a vector of velocities, meaning for each individual cell, not only what is the expression of a cell, but how is it moving? Is it moving up or down for this cell, for example? Then this may be a way to fit a dynamical system to predict the evolution as a function of the state, right? And this would give, for example, a gene regulatory network saying which gene expression controls the evolution of which other genes. So to some extent, it's possible, to do it. The difficulty is that we don't have access to velocities of each cell. We have access to the expression of each cell. So a question of how do you infer velocity is still a bit open. Actually, there's been very nice work published in the last few years on showing that in the data, there is some indirect way to assess velocity just by comparing the number of reads in introns and exons, etc. We proposed a different method, which is just based on, forget the equation, but just based on when you have a cell and in the middle of many cells, you look what are the cells around it, you infer what are the cells which represent future states, previous states, and you just make finite differences between them to infer the most likely direction of movement. So these are like this is a different way to infer velocity. And we were able to show that, trying to see if this method is able to reconstruct regulation, we're able to show that there is some signal, like on different data sets. Vertically, we have the AUC. So at 0.5, you would be bad, and many methods are bad, but our method is a bit better. but it's still very, very bad. Like the performance of how well these methods are able to reconstruct gene regulatory networks is, is not usable, because you're better than random, but not much better. So I think there could be much room for improvement here, and in particular in combining other types of data, probably using both single cell expression and epigenetic marks would be a better way, probably to try to infer the mechanisms underlying regulation of genes. All right, so I stop here. I'm sorry. I think it's 2 minutes after the hour, so it's just exactly 1 hour. I started 1 hour ago. I started talking. So I would just say, I mixed a bit at the end things which are not related to intratumor heterogeneity, but wanted to to come back to the initial point, which was that for cancer, at least, trying to better understand the complexity of tumors is a fascinating and probably useful question. We did some work on whole exome. I think that could be improved, but we reach a limit in bulk sequencing, and so I see really the field of single cell genomics. Like many people, I see it as a super exciting way to be able to decipher the complexity of the tumors. But it comes with so many questions, open questions, that if some of you, for example, want to work in that domain, I think there's going to be work for the lots of work for the coming year. So I'll stop here. Thanks for your attention, and I'm happy to take any question.
Hello, can you hear me?
Yes.
Yeah, so I'm Charles, a PhD student here. Very nice talk. So would you please go back to the sarcoma case that you showed the result for Clone-6?
Yes. So do you mean this one?
Yeah. So I have 2 questions here. Would the tools be able to discover the novel signature, the signature that has never been described before?
So currently not, because we decided. So we decided to start from a set of signatures. So to say, Alexandrov defined the signatures, and we decided to say what characterizes a subclone is that probably it uses a mixture of a few of these signatures. And this is a bit technical, but this means that in this graphical model, we have a UN variable which is just an indicator of what is the clone. So UN could be 1, 2, or 3 if you have 3 clones. And for each clone, we have SN, which is one signature among the 67 signatures or so. Right? So we decided to say that it's a signature. So each signature itself is a distribution of our mutation types. And so we say that given a clone, you may use different signatures with some probability, and given a signature, you create a mutation. So we decided to include these node using signatures just because we wanted, even to convince our colleagues that we started from non-signatures, and therefore, for example, we could do this analysis on TCGA to say signature 7 is overexpressed, signature 5 decreases, etc. This being said, if we didn't want to start from a known set of signature, it would be completely possible. In fact, it would just mean that SN would be considered as, not just a distribution over the dictionary of given signature, but that we could have other, like a dictionary to discover. We would say we can allow a number of clones and a number of basic signature, and just optimize the signature definition on the data. So this would be doable. We didn't do it, but it's doable.
In general, there should be, in theory, there should be 96 total signature, right? But not all.
No. So I was not clear. I think we have 96 types of mutations, right? So each mutation is one of 96.
Right.
Now, what I call the signature is a distribution over the 96 types.
Yep.
Right. So, for example, signature 2 is a distribution over that, etc. So we don't have 96 signatures.
The reason why I ask is, can you go back to the sarcoma slide? This is very similar to the situation I'm facing. So the type of cancer I study also show the same observation. There is a change in signature in a population, in a subpopulation. And when I plot the cancer cell fraction like this case, we also detect like almost like a bimodal distribution. And the signature of the subclone is very weird. Like it doesn't really look like anything normal. And so I think that this can be something very interesting in terms of biology. So do you have any suggestions to modify your tool so that I can kind of discover the novel signature?
Well, yeah, I know how to do that, but I would need probably someone working on it. I mean, it's not completely trivial. The reason why it's not completely trivial, I mean, if you want to know the technical part of it, is that when we fix the signature, then we just treat the samples one by one. So for each patient, we have whole exome sequencing, so we have a data sets, and we just fit this model to obtain for this patient the number of clones, etc. Now, if we want to infer the signatures, something specific is that the signature should be shared across all patients and all cancers, because this is really the fundamental notion of signature, is that different cancers use different signatures, but the set of signatures should be universal. So concretely, if you want, mathematically, it's quite easy to say, well, we just need to maximize the likelihood over the set of signatures. I can write the equation in one line, but to implement it, we would require some work, because compared to what we currently have, we need to change the code so that typically we handle not samples one by one, but we handle the whole TCGA in one shot and have parameter sharing across the different samples, etc. So conceptually, it's easy. Mathematically, it's easy to write it, but technically, I cannot promise to do it because it would require some significant work to optimize the signatures.
I see.
But if you want to work on it, if someone wants to collaborate on that, maybe we can think of that.
The second question is, what would be the minimum amount of mutation you would suggest to run these tools? Because I have a few cases, even when we only do exome sequencing at 400x coverage, I only detect around 150 mutations. Is that too low to do clustering?
No, so I think it's exactly what we target. 100, number of mutation depends on the cancer type. I suppose you're talking of whole exome sequencing if you talk of 100 mutations. So, on our simulations, but these are just simulations, but we tried up to 20 mutations, 50, 100, and on mutations, the performance is not as good, but it still works. And when we applied it to TCGA, so we don't know if the result is good, but at least we tried it on all samples, and at least the code runs and gives something. Probably one way to think about it is the only risk you take with this method is that if you don't have enough mutations, then it may miss clones just because there is not enough signal. Like, in the extreme, if you have a subclone but you have no mutation in there, you will not detect it. Now, if you have a subclone with 3 mutations, think of it in terms of this matrix, you will have 3 points a bit isolated, then there would be not enough statistical evidence for it. So on simulations, we have validated that this thing gives reasonable results as soon as you have, let's say, 100 mutations. So when you say I have 150 or so, I think it's worth trying.
Okay, thank you.
Great. May I ask next? It's Gil Oman. I'm a senior faculty member here. We're delighted you presented today, and especially our connection to Paris and Google. I was at Google Paris 6 years ago on a visit. I'd like to ask you specifically about applying your methods to circulating tumor cells, because one of the big challenges in tracking the heterogeneity of cancer, heterogeneity of all cancers, I think, in a clinical setting, is longitudinal. What happens over time in the patient? We can't sample an internal tumor periodically very practically, but we do have the capacity to pick up circulating DNA and circulating tumor cells. And then the question is, which of these methods are sufficiently sensitive to be useful in that mode? And if you have time for just a quick comment about your own career, I think some of our students might be fascinated to know how you currently tie together Google Health and Institut Curie and Simons Institute. three remarkable places. Maybe it's just one big collaboration. Maybe you do different things, different places. I think people be curious.
Okay. Okay, so for the first question on circulating cancer cells, yeah, so I think you're completely right that in practice at the clinics, I see that as a very, very promising way to have longitudinal follow-up of cancer, not just sample blood and sequence it. So I think what I presented, none of it, specifically designed to analyze circulating cancer cells. And I think what would be interesting in circulating cancer cells, if you follow them in time, as you suggest, would be to invent method, to check the progression, the evolution, the mutations in cancers, cancer genomes a long time. So this is probably none of the problems I mentioned, but I think it would be super, possible and super exciting to work on that.
Central.
So I'm sorry, I don't have much more to say, but I see that technically as one of the most promising way to, if we think of, especially I would say for me this is quite related to, maybe this is what you had in mind as well, to these plots saying, if we want to change a bit our way to treat the tumors, and instead of being a bit naive, big chemotherapy and then hoping there is no relapse, but sometimes the relapse may appear. If you want to have a finer control, a bit like what's done, in AIDS treatments, HIV treatments, where you follow the load in the blood and you follow the mutations, etc. Maybe one day we'll be able to, to accept that the cancer is still in the patient, but we could follow continuously, and maybe in cancer cells, circulating cancer cells, the mutations, and have alerts as soon as we detect the presence of mutations, etc. I don't know, is it something that you had in mind when you asked a question of this kind of possible applications of not only following the presence or absence of the tumor, but also following the evolution?
Oh yes, but there are methods now, very nice methods for extreme microfluidic devices that can pick up many different tumor cells and analyze for receptors, treatable, targetable receptors on specific cells. And quantitatively, there's a lot of progress in this domain.
Yeah, that's fascinating. Then the question about my own trajectory in time, and Google and all these things. So I had my own. So I've always worked both in machine learning and in computational biology. I've already tried, especially in the biology field, to be close to doctors and applications. So I had the position at the Curie Institute. I work with doctors, because I think this is one of the key. I don't know if I managed to do it, but at least I think that to be able to have some influence and to really work on the real problems, it's important to work in close contact with the experimental labs and doctors in this case to understand what are the problems. So this is what I did before joining Google. And the fact that in parallel to that, I worked on machine learning made that probably, Google was interested also in what I was doing in machine learning, and they contacted me to join their lab. So I joined Google when they started the lab in Paris. It's part of Google called Google Brain, which is, let's say, open research, kind of academic research, where faculties, we can do a bit whatever we want. Of course, they choose who they hire, but when they hired me, they told me, well, continue doing machine learning, and if you and continue doing comp bio if you like it, if you think there's potential there. What is clear is that, so I don't think there is like a plan, in Google to say let's solve cancer. It's not. We are much more modest. It's not easy, but at least the idea that the recent progress in machine learning, deep learning, for the moment mostly affect speech, language, vision, computer vision, etc. One question is, can we help more use these technologies to make progress in science? In this case, could be in biology, maybe in medicine. And what I suggested at the end is that this is, there are very active fields of research trying to use the latest machine learning approaches to analyze these data. So this is a bit what I try to develop at Google now. But again, Google, there is no wet lab. there is no real wet lab except maybe in some lab. Google is so big that probably there are some wet labs, but let's say it's not like the Curie Institute. So to be able to work on that, we still have, we work with collaborations. This is very open research. We publish everything, all the code, all the papers. So, for example, in Paris, I started this activity, and we naturally, because I was at Curie Institute before, we started a collaboration with the Curie Institute. We do have some PhD, one for the moment, one PhD student. I mentioned Felix here. So this is like an official collaboration between Google, Curie Institute, and Felix. And for me, it's super important. So here we collaborate with a colleague, Céline Vallot, who has an experimental lab on single-cell epigenomics. So she developed the technologies. We work with the samples from the hospitals, etc. And the fact that it's Google versus academic for the moment doesn't make a big change, except that we don't have to ask for grants outside. We have enough money to fund the collaboration. But for me, so Google AI, Google Brain is very much into these open academic collaborations, and clearly we see the collaboration with Curie as one example of the collaboration we need to work in this domain. Like we don't have data, we don't have expertise in cancer omics, so we're very happy about that. And then you mentioned, I think, the Simons.
Yeah.
So for the moment, I mean, personally, I'm not aware. I'm not involved in any collaboration between Google and Curie and the Simons. I did in the past, when I was at Berkeley as a sabbatical. I was also hosted by the Simons Institute for a while, but this is a personal experience. But overall, I think Simons is obviously doing lots of exciting things, and there may be some projects I'm not aware of between Simons and Google, maybe, but I cannot comment on that because I'm not aware of it.
Well, very good. Shows great opportunities for our young people.
Yeah, I think so.
Can I have a quick question? That was a great talk. You had that picture of how the tumor grows, you get more heterogeneity, and then you have chemotherapy, and you go through this kind of bottleneck. And so when you then measure the profile of the tumor that grows out, is that simpler? Is it less heterogeneous than what you started with?
So I would say yes, but if you challenge me, I don't have much evidence. So I would say yes because this is what I remember, heard from my discussions with my colleagues, and so we decided to work on this. Like even in this paper, I think that I show here, they discuss that. But overall, I mean, at least at the beginning, overall the mechanism that is expressed in this publication and that reviews several of the publications is that things that happen mimic a bit what we observe in controls of populations in ecology or in pandemics, etc. Is that you have heterogeneity. Then when you have some event that kills most of the people in the species, then some survive, and you have the start of a new population, which initially is much less heterogeneous. So I think this is the message in this. I mean, it's logical, and it's also the message in these publications. Even though I cannot comment on the evidence that support this.
Just another question. If you think of, you looked at mostly mutations, but if you look at structural variations, chromosome rearrangements, what's the heterogeneity look like there? I thought those are kind of early event that leads to clonal. They're clonal, so you find them in all the metastases and so forth.
Oh yeah, so I think it's very debated, and in particular I see it, when for example, when we have submitted a CloneSeq to review, and we are still discussing with the reviewers who, some of them don't accept the fact that we can just rely on mutations and say, probably the most important part of this heterogeneity after clones start to evolve is accumulation of copy number variants, of structural rearrangements, etc. So as far as I understand, it's debated. Like there is not much evidence that it's typically first mutations, then structural changes. I think Many people thought that the opposite is more correct, like we mostly have early structural variants and then mutations. But there are many evidences, many cases where there's evidence that we have subclonal, for example, copy number variants, so like the rearrangements that change after subclonal events, etc. So I'm sorry, I don't have a clear picture, and I'm not sure everybody, people don't agree on that at least, but it's certainly not simple. And looking at the mutations is in a sense super naive, super simple to explain the complete sorting. Mostly first, we see it as one way to quantify, one marker, one signal that we can measure that help us make this picture here. But inside of this picture, maybe each clone should be characterized not only by mutations, but by maybe other things if you want to understand their properties, their logical properties.
I have one
Thank
question
you.
that I
Thanks
think
so much.
the rest
Very
of
good.
that I think maybe several individuals in the audience might find interesting. Are there actually plans within your groups to move beyond exome sequencing to actually look at non-coding regulatory elements like super enhancers, some of these other sort of genomic features that have been tightly correlated to a variety of sort of cancer subtypes?
Yeah, of course. I mean, we would like to work on it. I mean, many people work on it. It makes complete sense, yeah. What I can say is that in our case, so I said we started a project with the Curie Institute on breast cancer. And there we're specifically interested in epigenomics changes at the single cell level. So in this case, we look at the histone modifications. So it's not only not only DNA, but also beyond DNA, how the structure of DNA changes, how the regulation changes. And it's clear that we work on that because our collaborator in Curie is focusing on a specific subtype of breast cancer where it has been shown that resistance to chemotherapy is driven by the emergence of subpopulations of cells with different epigenomes, different histone marks. So we work on that, and it's clear that it seems to be relevant.
Thank you.
Can I have ask a question?
Yeah.
Do you hear me? Okay, great. Thank you for your presentation. I wanted to ask about the methods of your tool, specifically asking about the calculation of CCF. So in some of the cases, you have your CCF above 1, and it can happen because of different type of calculation of CCF. So first of all, I want to ask what is your method, or how did you calculate the CCF? And secondly, is that how do you handle the CCFs above 1?
Yeah, very good point. And I'm sorry, I will not give a good answer because I forgot. I didn't do it, so most of the work was done by Judith, the PhD student, and in this case, I just forgot exactly what tool she uses. I mean, it's not a tool; it's an equation, right? It's in the paper. Mm-hmm. Now, the fact that it's above 1 is just, as far as I remember, it's just the result that we use. So we use an equation that basically transforms the raw frequency. So, for example, you have let's say 80% of the reads which are mutated, and combines this number with another number, which is the estimated proportion of cancer cells in the tumors, right? And so if there was no noise, the simple equation would be that more or less like if you have 80% of mutated reads and you know that you have 80% of cancer cells, then the CCF would be 1, because it would mean that all the mutations are in all the cancer cells. So somehow you have a fraction, right, that corresponds to 1 when you have the percentage of mutated reads corresponding to the estimated fraction of cancer cells. And the fact that it goes above 1 is because we have, in a sense, a bad estimator, which is that if we have more, so in proportion, more mutated reads than number of fraction of cancer cells in the biopsy, then our division gives something larger than 1. So I would say we know that the CCF cannot be larger than 1, but we just use this estimator, which is outside of the range, because it's a better estimator when we think in terms of modeling the true value plus some noise, I would say.
True.
If you see what I mean.
Yeah, I totally understand. And like as a second part to that question is there any like rigorous quality controls that you have on your results after finding the subclones and the peaks in the figure that we are seeing? Is there any rigorous quality control that you do? Any possible pitfalls that you look for?
Nothing specific. So I would say that, the whole pipeline is a bit, I mean, there are a few steps, like typically to infer the number of peaks. So what we do, to clarify a bit, we have this graphical model here that we estimate by maximum likelihood, and then we estimate the number of clones by some model selection criteria, etc. So there is some, process there that we have optimized, tested, so that we think our estimates are the best possible. Just to this is a bit more complicated than just fitting Gaussians on this picture, even though conceptually it's doing the same, but it does it in the space of parameters, etc. So we say there is no specific quality control or thing that we do, but let's say the processing, like the choice of the model selection criterion, to say, we compare one clone or two clones or three clones in terms of likelihood, and we check, we do model selection. So this thing has been a bit optimized, so that on average we have the best possible output.
Okay, thank you.
You can call that a kind of quality control and post-processing.
Thank you.
Okay, do we have additional questions from the audience? If not, let's stop here because we are really, really over time today. It's really, really rare. We have today like
I'm sorry,
like almost half hour overtime. But that's a sign like your talk is really, really good. Thank you so much, Jan-Philippe. So I think you sign up like to talk with a few trainees. I think they are probably still online. Let's just do a little bit until you have to leave. And Aaron, you can stop recording.